Hey everyone, and welcome. Today, we're going to dive deep into the DNA of things like plants and fungi, and we're going to uncover these secret assembly lines that life uses to build some of its most powerful and, frankly, amazing molecules. This is the kind of stuff that's actually rewriting the biology textbooks, so let's get right into it. For the longest time, the rule in biology class was pretty simple. In simple life, like bacteria, the genes are all organized in these neat little toolkits called operons. But when you get to complex life, we're talking eukaryotes, like plants, fungi, and even us, the genes just seem to be scattered completely randomly. It's like trying to bake a cake, but in bacteria, all your ingredients are on one shelf. In our cells, it's like they're tossed all over a giant chaotic library. But what if that whole chaotic library idea isn't the full story? What if, hidden in all that mess, there are these secret, perfectly organized shelves? That's the exact mystery we're going to unravel today. Well, it turns out the answer is a huge yes. Scientists have found this incredible exception to the rule, and they're called metabolic gene clusters. These are actual sections of DNA where all the genes needed for one specific chemical assembly line are physically lined up, right next to each other, ready to go. It's a level of organization in complex life we just didn't think was there. It's a real game changer. Okay, so before we really dig into these clusters, we need a quick primer on how a cell's chemistry, its metabolism, works. You can pretty much split it into two halves. Think of it as the must-haves for staying alive versus the specialty tools for unique situations. So on one hand, you've got primary metabolism. These are the absolute essentials for life. Things like basic sugars and amino acids that every living thing needs to build itself. But then you've got secondary metabolism. This is the specialized stuff, poisons for defense, colorful pigments, or even powerful medicines. And this is exactly where these gene clusters become the star of the show. All right, so let's start our journey with fungi. For quite a while now, scientists have known that fungi are basically the undisputed champions of this gene clustering strategy. Their DNA is packed with these dense little groups of genes that act like tiny, super-efficient chemical factories. And you know the most famous product to ever come out of one of these fungal factories? Of course you do. It's penicillin. This antibiotic that literally changed the world isn't just some random fluke. No, it is the direct, intentional result of a dedicated genetic assembly line inside the Aspergillus fungus. Okay, check this out. What you're looking at right here is the actual genetic blueprint for making penicillin. You see those three main genes, ACVA, IPNA, and ATA? They are the core recipe, and they're lined up right next to each other. You can think of it like this. ACVA starts the process, IPNA does a key modification, and ATA puts the finishing touches on it. The whole recipe, all in one spot, it's just perfect. And this is what you get at the end of that perfectly organized assembly line. From that one little cluster of genes, the fungus produces this incredibly complex and life-saving molecule. It's such a clear link between organized genes and a world-changing chemical. Okay, so from fungi, let's jump over to the plant kingdom. Now, for the longest time, everyone kind of thought this clustering thing was just a fungal trick. But more and more, we're finding out that plants are doing it too. Their genomes are also full of these organized clusters, acting like nature's own little pharmacies. But then, we found stuff like this. Get a load of this. This is a gene cluster from the opium poppy that makes a medicinal compound called noscapine. And we're not talking 2 or 3 genes here. This is a massive 10-gene cluster. All 10 of the enzymes you need to build this complicated molecule are sitting right there, side by side in the DNA. This was huge because it proved that this complex clustering strategy wasn't just for fungi. It's used across the kingdoms of life. Okay, so we've seen that these clusters exist in both fungi and plants. But that just leads to the biggest question of all, right? Why? What's the evolutionary advantage? Why go to all the trouble of keeping these genes physically together instead of just leaving them scattered all over the place? Well, first off, it's cool to know they don't all look the same. They come in a few different, let's call them architectural styles. You've got your all-in-one cluster, like what we saw with penicillin. Then there's a core and satellites model where you have the main group, plus a few helpers elsewhere. Sometimes they're even intertwined with other pathways. And in the most extreme case, gene fusions. You have 2 genes that literally merge into one giant multitasking protein. So why bother with all this organization? Well, this quote right here really nails one of the biggest ideas.
By keeping all the genes for one pathway physically together, it's way, way harder to accidentally lose one of the crucial steps during the genetic shuffling that happens over generations. It keeps the whole factory intact.
Let's just break down the three main theories here, because they're pretty cool. First up, co-inheritance. Think about it. If all the tools are in one box, it's much easier to hand the entire working toolkit down to your kids. Simple. Second, co-expression. It is so much more efficient to flip a single on switch that turns on the whole factory rather than having to run around and turn on a dozen different switches all over the genome. And third, and this one's really important, toxicity. Sometimes the steps in these chemical reactions create toxic byproducts. If you only have half the assembly line, those toxins can build up and kill the cell. Clustering makes sure the whole process runs from start to finish safely.
This is way more than just some cool biology fact. This has massive real-world implications. Because now that we know these clusters exist and we know what their signatures look like in the DNA, we can start a totally new kind of treasure hunt. We can literally go mining for genetic gold. And the potential here is, well, it's absolutely staggering. Just look at this number. By scanning fungal genomes, scientists are predicting there are over 10,000 of these gene clusters just in fungi, waiting to be explored. Each and every one of them is a potential factory for some new, unknown molecule with who knows what kind of properties.
And this modern-day treasure hunt has a name. It's called genome mining. Scientists use these incredibly powerful computer programs to sift through mountains of DNA data, looking for the telltale signs of these clusters. It's a way to predict what amazing new drugs or antibiotics or even new materials an organism could make before we've even seen the chemical itself. And all of this leaves us with one final and honestly pretty thrilling question. For thousands of years, we found our medicines by just testing stuff from nature. We were basically working in the dark. But now, by understanding these secret assembly lines in DNA, we can search with a purpose. So what incredible new medicines and materials are still out there, hiding in plain sight, just waiting in the DNA of the life all around us?